Good morning, everyone. My name is Zhang. I'm a postdoc in Dr. Shuo Xiao's lab from Rutgers University. And today, first, I would like to thank the committee for giving me this opportunity to present my work here, which is about the vitrification preserves follicle and oocyte functionality in an encapsulated in vitro follicle growth, or eIVFG system. First, you may be wondering about why we want to do the individual ovarian follicle vitrification. So currently in our lab, we have multiple projects going on, including the reproductive toxicology, organ-on-a-chip, non-hormonal contraceptive discoveries, and of course, the Oncofertility related studies. So most of the studies will heavily rely on the ovarian follicles. And for example, we have a project that we study the ocean environmental contaminants effect on the dolphin's health, where we collect the dolphin ovarian follicle samples and perform the toxicology studies. And you can imagine that unlike the mouse or laboratory species, it's almost impossible for us to get the dolphin ovarian follicles at any time as we wish. So it's really important for us to vitrify those ovarian follicles when we have access to them and build a biobank. And also, you may echo a talk given by Dr. Anderson yesterday, and he mentioned that for the ovarian tissue cryopreservation, for the cortex part, which contains most of the primordial follicles, it will get sliced and frozen. And for the remaining medulla region, with a lot of COC and oocytes, we could potentially do the in vitro maturation. Then we want to ask, how about the remaining growing follicles? Is there a way we could vitrify those growing follicles, and which could potentially provide us with a new approach for the fertility preservation? Vitrification is a commonly used cryopreservation method, and it has been widely applied to the oocyte and the embryo in the clinical settings. And however, it could be totally different stories when we want to vitrify the follicles, as the follicles have a much more complex structure, and one of the most challenging part would be how to ensure the cryoprotectants were well permeabilized and also distributed evenly to make sure we can avoid the direct cell damage or also the damage caused by the loss of cellular connections, for example, the gap junctions. So in this study, we collaborated with Dr. Mary Zelinsky and developed this closed vitrification system for the individual follicles, and with the optimized vitrification recipes. And with this whole system, we could achieve a more than 95% survival rate post-vitrification. We combine this follicle vitrification system with our encapsulated in vitro follicle growth follicle culture method, or eIVFG. So with this eIVFG culture method, during 8 days of culture, the multilayer secondary follicle could develop into the antral stage. And when we induce the HCG stimulation, the antral follicle could ovulate ex vivo. By applying the eIVFG method, we could see that both the fresh and vitrified follicles could develop into a fully matured antral stage during 8 days of eIVFG, there's no significant difference for the follicle diameter throughout the culture period. And in addition, there is no significant difference for the estradiol secretion in the conditioned medium from both vitrified and fresh follicles. Let me see if I could play this video here. Oh, sorry. Do you know how I could? Or if you can help to click on the center one, the up center panel. Yes, thank you. Yeah, here we go. Yeah. Cool. So here, there's a time-lapse video showing after the HCG stimulation, both the fresh and vitrified follicle could respond to the HCG and start to expand. And finally, one side of the follicle will rupture, just as what happened in vivo. And also you can see the cumulative cells are expanding out. So we're also collecting collected the metaphase 2 oocyte from the ruptured follicle and then stained them with the alpha-tubulin antibodies. So on the right-hand side, you can see for the vitrified follicles, they have a similar M2 percentage compared to the fresh ones. And you can also see the image on the top right panel showing that they display the normal spindle morphology as well as the chromosome alignment. So now we have demonstrated our vitrification system combining with the EIVFG could preserve the follicle functionality, including the follicle development, hormone secretion, ovulation, as well as the oocyte meiosis. Then we want to ask a question, how about at the molecular level? So to answer this question, we performed the single follicle RNA sequencing analysis. So first, we start with the antral follicles that we collected on the day 8 of EIVFG and performed the single follicle RNA-seq. By the PCA plot, you can see the transcriptome of fresh and vitrified follicle. They are highly overlapped, suggesting they're very similar transcriptome profile. And on the down panel, you can see some representative follicle maturation-related genes. Their expression levels are also very similar between the fresh and the vitrified groups. For example, the CYP19A1 or aromatase, which is responsible for the estrogen synthesis, and also the FSHR, which is an important hallmark gene indicating the follicle maturation, as well as some CICA cell marker like the CYP17A1, and also the oocyte-specific genes, GDF9 and BMP15. So now we have demonstrated again the vitrification could preserve the molecular signature of the FSH-dependent follicle maturation process. So next, we want to look at the LH-dependent ovulation process. Similarly, we collect the single follicle samples by a time course collection of 0, 1, 4, and 8 hours post-HCG stimulation and perform the single follicle RNA sequencing again. So similarly, by the PCA plot, we can see the fresh and vitrified follicle were highly clustered together based on their collection time. But within each of the corresponding collection time, these two transcriptomes were highly clustered together. Also, the heat map, you can see their gene expression patterns are also very similar between the fresh and vitrified group. I also show some representative ovulation-related genes, including the AREG, PGR, PTGS2, as well as the STAR. Those are all important gene markers for the ovulation process. And you can see there's a very similar gene expression pattern between the fresh and the vitrified group. So now we have demonstrated for the both FSH-dependent follicular genesis and the LH-dependent ovulation, our vitrification combining with the EIVFG could preserve their molecular signature. Then next step, we want to look at the oocyte or oogenesis. It has been reported that there is a degradation of the maternal mRNA during the oocyte meiosis process, which is critical for the future development stage, embryo development stage. Therefore, we collect the M2 oocyte after the HCG stimulation from both vitrified and fresh follicles and perform the single oocyte SMART-Seq2 technology. And here is the result. First, the PCA plot shows again the transcriptome from the M2 oocyte from both vitrified and fresh follicles. They are highly clustered together, suggesting their high similarity with the correlation coefficient R value equals to 0.99. And I also show some representative oocyte marker genes, for example, the CP2, GDF9, as well as the BMP15 here. You can also see they have a very similar expression pattern between the fresh and vitrified follicles. We also further compared 84 reported or published maternal effect genes. So within these 84 genes, there's a very high correlation coefficient R equals to 0.996, suggesting there are very similar expression pattern between the fresh and vitrified follicles when we compare the maternal effect genes. And here I also have some representative genes listed here. Good. Oops. So with that, I would like to conclude that our closed vitrification system combining with the EIVFG could well preserve the functionality of follicle and oocyte. And it has multiple applications besides the fertility preservation. For example, the conservation of the endangered species, or as I just mentioned at the beginning, we can perform the reproductive toxicology studies and drug screening, as well as other ovarian biology studies. With that, I would like to thank my advisor, Dr. Shuo Xiao, and all my lab members. I would also like to thank my lab members and our funding support. And thank you for your attention, and I will be happy to take any questions.
Thank you. Great, we have time for one question. Dr. Silver.
That was just a spectacular presentation.
Thank you.
It was really spectacular. I just have a simple question. When we freeze ovarian tissue with vitrification, we have to use much higher viscosity, like 20% DMSO, 20% ethylene glycol. What was your cryoprotectant for freezing whole, entire, but individual follicles? Was it higher than the usual?
Yeah, thank you for the question. So actually our recipe, our vitrification recipe contains about both DMSO and also the isoglycol. And they are at like relatively high concentration. We have like 3 different concentration series, starting from 0 to 15% and then ultimately to 40%.
Ah.
Yes.
Fantastic. I really applaud your work. It's great.
Thank you.
Thank you, Dr. Zhang.